Perfect. Well, welcome everybody. I see we already have a few attendees having joined already. So we are pleased to bring this next installment of our Kickstart Presents webinar series. The title of today's talk is From Idea to IND, Tips for Successful Early Product Development. And this is a question that we gotten from a lot of the startups we work with, who are interested in really, getting, the process started on their own therapeutics or devices, and they want to understand what it takes to submit their applications to the FDA. And in this case, we're going to get very valuable insight and reviewer, ex-FDA reviewer insights into key factors for successful IND submissions. And we have a fantastic panel today from Parexel, who is a global leader in biopharmaceutical and contract research organization services that provides tailored services to help biotech and medical device companies, like many of our startup companies, to meet their development goals. And it is headquartered in Durham, so here in the Triangle, and has more than 700 employees, including most of their C-level leadership and CEO Jamie McDonald. Parexel is focused on supporting their partners to develop innovation therapies to improve patient health, and they do this through a suite of services that help life science and biopharma customers across the globe transform scientific discoveries into new treatments for patients. They were founded as a regulatory consultancy over 35 years ago and have evolved into a full-service clinical and drug development solutions provider. Again, today we have a great set of panelists, and I'm going to start by introducing Julie Stover, who is the Senior Director of Regulatory Solutions from Parexel. She has over 27 years of clinical development experience, and 7 of those have been with Parexel in the last 7 years. She leads the North American team of regulatory strategy and solution consultants that focus on helping biotech companies, again, like many of the ones here today, in navigating the regulatory and payer landscapes to achieve the fastest, most efficient pathway for developmental goals. Julie will also introduce some of our other panelists today, so Partha and Mo. So Julie, please go ahead and introduce them.
Great, thank you very much. Very excited to be here today. As Maria mentioned, we are a global contract research organization. I think she mentioned 700 in North Carolina. We are 18,000 globally. But we did start as a regulatory consulting agency, and really excited to be able to have 2 of our subject matter experts to talk to you guys today. Both Partha and Mo have extensive experience working with biotech companies. Just like you guys, we work with companies that are as small as one and as large as the big pharmas. Specifically today, we've got Dr. Partha Roy with us. Partha has 19 years experience in drug development with a specific focus on clinical pharmacology. He's been with us at Parexel for the last 8.5 years. Prior to that, he was a clin pharm reviewer in the FDA's Division of Pulmonary Allergy and Rheumatology. Partha's focus at Parexel now is as a regulatory strategist, and really helps biotech companies focus on the full spectrum of pre-IND all the way through to NDA and or BLA filing. And I can tell you personally, Partha is my go-to expert for all things U.S. regulatory strategy. We also have Dr. Mo Hadaran, who has joined Parexel, and forgive me, Mo, I think it's 2 years. He'll correct me if I'm wrong. Joined us straight from the FDA as acting branch chief of OTAT, the Office of Tissue and Advanced Therapies, where he worked for 8 years. Mo has a specific focus on CMC, so chemical manufacturing and control, both from early CMC considerations as well as manufacturing and facility inspection. And Mo is known as a CBER-wide subject matter expert and has been integral here at Parexel in the development of our cell and gene therapy working group, which is a multidisciplinary team of specialists dedicated to those types of therapies. So really excited to have both of them with us today. I think if we want to jump into the slides.
Yeah, that'd be great. And I just wanted to tell everybody that we have some questions we'll be asking after the presentation to the panelists. And Judy Prasad, our Kickstart Operations and Program Manager, will be collecting as well during this presentation any questions that the audience has as well. So please go ahead and write your questions in the chat and we'll collect them and ask them at the end. I'm also very, very excited to say that Parexel can have also office hours with UNC startup companies and faculty interested in speaking with them as, about their own projects. So we'll be sending at the end of the webinar a link to all the participants who are interested in registering for it. Thanks, and let's go ahead and start the presentation.
Great, thanks. Just a couple of introduction slides before Partha takes over, and I'll move quickly because he's the main event, him or the main event. But just wanted to note that, you know, although it does seem that big pharma seems to get the lion's share of press and exposure, we here at Parexel have really recognized that biotech companies such as you guys are where all of the innovation is coming from. And, you know, this data here really demonstrates that although big pharma is obviously a big player, if you look over on the right, 81% of biotech companies like you own, or biotech owns 81% of the overall pipeline of development. So definitely a recognition that you guys are on the leading edge and where all of the innovation is. And the bottom stat really shows that over the past 20 years, investors and funding are paying attention. We've noticed a 179% increase in the amount invested in venture financing in the past 20 years. So definitely a shift in industry on the focus on biotech. And we here at Parexel have done that as well. This team that you're talking on the phone or hearing from today are specifically focused on working with biotech companies. So if you want to move on to the next. Just a little bit of information about Parexel Consulting. I think we mentioned we're an 18,000 total employees, with 1,000 of us really focused on regulatory globally. We've got 100 or roughly ex-regulators and health technology assessors. You've got 2 that are going to be speaking with you today, and we cover 110 different countries. So regardless of where you want to go in the world, we have the regulatory experience to help assess the right pathway and help you get there as quickly as possible. Next slide. Just some highlights, I'm not going to read all this to you, but areas that we help clients like you in trying to understand where you are from early preclinical development to where you want to go, whether that is taking your compound through proof of concept and selling it off, or if you have aspirations of taking it all the way through to marketing yourself. We have exposure not only to the regulatory side, but also focused on how to help you understand what your data package looks like and how to position that in the best way to achieve funding to get you to your next goals. And really what we try to do, and my particular role is that bottom right hand, which is identifying specialists with the right technical and therapeutic expertise to augment what your team looks like and help you figure out the gaps to get you on to the next phase. So how do we help you get from the concept that you have to the IND, into the clinic, and get your compound in the hands of patients? And with that, I'll hand that over to Partha and let him talk about some of the steps that we use to get there. Partha?
Thank you, Julie. And thank you, UNC team, for the opportunity today. So good afternoon, everybody. In the next 12 slides, hopefully, me and Mo, have these slides. The intention is to have the Q&A session at the end so that you can ask questions, and wanted to make sure that you have enough time to do that. So as Julie mentioned, some of us have been doing this particularly with the regulatory agencies around the world and working closely with them and actually working with the biotech industry and bringing them together in different types of interactions, engagements, IND, and up to the entire development pathway. So I'm gonna speak a little bit from the CDER side. I mean, as you may know, that the CDER regulates drugs including monoclonal antibodies and peptides and some of the proteins. So I will talk about the CDER side because my experience is specifically on that. And I think Mo will cover the CBER side of things. So anyway, so this is the first slide, and you probably are very aware of getting from a research and development to preclinical testing to probably the IND submission, and then you have an IND action after a 30-day review, and then you're off to races in the clinical development. But before all of that, I think there are a lot of questions, how we can engage FDA, what are the different studies that we need to run to have a meaningful conversation with the regulatory agency, and how we can help, and what is the timeline of it. So all of that are very relevant questions and pertinent questions. Some of it is, of course, depending upon your particular development, type of products you're talking about, what kind of investigations that you are already doing, and where you are really at that stage where you can have a meaningful conversation. So in this slide, you see here, I put together a couple of interaction points with the FDA. For example, interact meeting, which I think Mo probably will expand because that's a very CBER specific meeting. It's an informal interaction, very, very early in development, trying to get an informal discussion. But most commonly, you probably will hear is a pre-IND meeting, which is pretty much both with the drug and the biologic side, and that's probably for most development, that's probably the first interaction with the FDA. And then after that, you prepare your IND and do an initial submission, and upon which you will have an action following a 30-day review. So next slide. Now, I was about to say that, there are a lot of questions around that. Should I submit a pre-IND before an IND submission? And oftentimes I usually say that, yes, absolutely, you should be doing that. FDA a lot of times is a very is a stakeholder and also very collaborative in nature in trying to help particularly small businesses, moving their drug development forward. The pre-IND is, between the sponsor and the review division, is a consultative mechanism, and really a mechanism to receive early feedback and guidance on the information necessary for a complete IND submission. It involves, few steps with a meeting request letter and a briefing package that goes to the FDA. You probably have to prepare quite a bit of before the actual meeting in terms of, rehearsing and who is going to speak what. Then, of course, the actual meeting preparation, and finally the debrief. Typically, such meetings are done about 3 to 9 months before a planned IND submission. It is often difficult to get a second meeting within a year, although there are cases where the sponsors has been able to secure the second meeting if the development path completely changes from the previous one. In CDER, the review divisions are organized generally along therapeutic class, and increasingly the pre-IND meetings has taken a written response only format. Obviously now, because of COVID-19, there is no face-to-face meeting for the last few months, and probably is going to continue to the end of the year at least. Then it is some of them important meetings are turning into a virtual meeting. And then there are some which are teleconference, but an overwhelming majority of the pre-IND meetings in CDER have become a written response only. Next slide. So now what is the process? You send the meeting request letter, you send the briefing package, then FDA provides a preliminary responses to the questions ahead of the face-to-face or a teleconference sponsor meeting. As I said before, a lot of the times it's a written response only. It is a good practice to let FDA know the questions that needs discussion in the actual meeting, and also do provide sponsor rebuttal as appropriate. Sometimes we need to prepare a small slide deck, and it's probably worth preparing to deliver some key messages and or describe and explain specific data. But often the regulatory project manager at the FDA, he or she is your friend, and you know, you have to let him or her know your intention ahead of time and submit the slide materials the night before the meeting. And then you have the actual meeting. I mean, it's oftentimes, a lot of times it's a face-to-face or a teleconference or a virtual meeting, as I said. And then, of course, you come back and do a debrief. And debrief is extremely important, so that you collect all of the information. And sometimes you'll be surprised that a lot of the times, you know, people go to the same meeting, they have a completely different interpretation, you know, what the agency has said or what the key points of the discussion. So it's very important to debrief properly. That also helps with the final meeting minutes. Next slide. So here is, you know, I mean, for the last many years, you know, I've done many of the meetings, both from the sponsor side and also from the FDA side, sitting on both sides of the table. So a lot of the times, you know, the sponsors get focused on whether we get an agreement for the agency or not. And particularly in the first meeting, a lot of the times you may not get that agreement. Oftentimes, the goal should be that, and does the agency tolerate your approach? The questions which are easy to deal with, which are straight yes and no, it's better to get an agreement quickly, whether yes or no, and then you kind of move on to the complex and the challenging questions. And really the art is to let the agency talk, because if they talk more, they actually will reveal to you what they're thinking while they reviewed your package. And oftentimes some of the questions, you know, it's more complicated than just a yes no answer. Oftentimes they will say, we do not agree. Their responses usually start with that. If they do not agree with any part of that question, they will start with that response. And not agree doesn't always mean that there is a disagreement. Oftentimes, you know, the question is probably complicated. Probably, you know, at that time they cannot really answer. They cannot give a comprehensive response, and oftentimes the question is beyond the authority of the reviewer that are responding to your question. So again, it is important that when you have a no answer, then it's a good strategy to kind of engage the agency and ask for the pros and cons, other possibilities and alternatives. That's why I put here at the bottom of many disagreement is a value judgment about what is really reasonable, and at a reasonable, what is at the reasonable. It might be unreasonable at a time when you are meeting. It might become reasonable as the development progresses. Next slide. So here are the key components of an IND, and as part of the IND regulations are in 21 CFR 31220 subpart B. And here are the three very core functional areas that comprise of the IND. So CMC, non-clinical, and clinical. CMC obviously involves analytical characterization. It talks about the manufacturing and the product quality. Non-clinical is often comprised of the mechanistic and potency issues and a lot of pharmacology and toxicology studies and investigations. In an initial IND, and oftentimes when you're trying to open an IND enabling first in human study, probably the clinical information is pretty slim. So the clinical protocol becomes the most important part of the clinical components. And if there is a previous human exposure or human experience, that needs to be a part of the clinical. Next slide. So here are the two types of, there are the types and the categories of INDs. Now the question oftentimes is which one is right for me? And obviously the answer is it depends. So the two categories of INDs are commercial and research. So most of the INDs that we see is commercial. You know, somebody is trying to, one of the commercial entities trying to develop a product in the particular indication for a particular target population. Research IND, which is non-commercial, the intent is not really to develop the product for final marketing authorization, but oftentimes it's initiated by investigators. And they want to do sort of the ground research on a product which may have been approved for another indication, but you want to do some research on something else, you know, and oftentimes for their own patients. So the types of INDs, you know, there are three sort of a broad types, you know, sponsor initiated, and the sponsor can be a company or an investigator, as I told you. Emergency use IND, that is oftentimes a single patient basis. Somebody is in a life-threatening or a very serious situation where they cannot wait to get their proper IND developed. You know, so there might be an emergency use IND provision. And then the treatment IND, which is a compassionate use and expanded access. Oftentimes, those two words are interchangeably used. In the regulations, there is nothing called compassionate use. It's always expanded access or a treatment IND, where you know, you are providing that treatment for a single patient to a group of patient population. You're expanding the access, but the important part of that is that that particular product is still being developed fully developed for marketing authorization. And the very seldom used is called an exploratory of phase 0. It has been there in the books for a number of years, but for probably a good reason why people haven't used it that much. Oftentimes, when the companies do screening of drug candidates, you can start a human study, mostly for you know, drug ADME property of the drug, you know, related to small molecules. You can actually go and do a study for a sub-therapeutic dose of maybe a number of candidates when you're trying to screen the candidates to see whether they have a property to become a drug or not. So sometimes, you know, the sponsors have used that. Next slide. So again, I don't spend a lot of time on this one. This is a typical ECTD or Electronic Common Technical Document. And here is a lot of the information that I already talked about. And I'll explain a little bit more. But it's really those three core areas: quality, that is CMC, non-clinical, and the clinical. And this ECTD backbone, that goes through the product life cycle. I mean, you submit that as a part of IND, and then you follow the same structure as you develop the product and finally going to an NDA or a BLA. Next slide. So what are the information that is important for each of those functional areas? CMC information, and I know Mo is going to probably expand on this quite a bit in terms of cell and gene therapy, but it's really to ensure the proper identity, strength, quality, and the purity of the investigational drug. The amount of CMC information will vary within the different clinical phase and the proposed duration of the investigation. the dosage form, and the amount of information otherwise available. Oftentimes for small molecules, it's two components, drug substance and a drug product. You have to give the FDA the information about the placebo, the testing and the release, investigational drug labeling, environmental analysis, or a request for categorical exclusion. All of these things are important part of the CMC information. And sometimes you have a combination product, and then you have to provide some of the information on the device side also. You have to provide information on storage and transport conditions and containers. Next slide. Now, another important part within the IND, when you go to the IND, is a non-clinical information. Now, how much animal studies you have to do, and what kind of information you need to produce to have a successful IND. So, adequate information about the drug's pharmacology and toxicology, but to support their use and a discussion of the rationale for the drugs development, intended dose, duration, schedule, and route of administration in the proposed trial. So when you're opening an IND, you're essentially asking FDA to provide you the permission to go into the to execute that IND protocol. So all of this information within that protocol is going to be supported, has to be supported by non-clinical information. You need to establish the mechanism of action, optimize pharmacologically effective doses and the route of administration, and gather some of the ADME information of the investigational product, if known. And I know in some of the products, the ADME information probably is not going to be relevant. Toxicology information comes in different buckets, acute, subacute, and a chronic toxicity study, and sometimes they run. You have to run a special toxicity studies, and sometimes it has to be route specific if it is non-oral routes. The kind, duration, and the scope of the animal and other studies required will depend on the duration and the nature of the proposed clinical investigation. Now, sometimes the million-dollar question is, do I need to be GLP? Do I need to be GMP, my product? And I can address that, and I'm sure Mo will probably expand on that a little bit more. But oftentimes, you have to remember that at the IND stage, it's the agency and everybody's very focused on the safety. So anything that you do in terms of safety pharmacology and toxicology studies, oftentimes the agency is going to expect as a GLP. Next slide. Now the clinical components, as I said, oftentimes if you are running your first in human study, then obviously there is not a lot of previous human experience. But there are many times when the drug is brought into the US from outside the US, and then you may have a lot of clinical experience. So that information that needs to be put in. So at the IND stage, as I said, it's about safety, and and the perception of benefit. It's only a perception at that point. So the agency is going to be very focused on the safety of your product. So that's why in your IND opening protocol, you'll have to get into the number of patients, the doses, regimens, safety exclusion criteria, stopping rules, safety monitoring plan for staggering the subjects. Informed consent, FDA oftentimes now are requesting to see. You also have to provide a general investigational plan, and the minimum requirement is 1 year. And then oftentimes you will see investigators brochure, and that's really a guidance for the investigators, but you have to define, at least at the time, you have to put forward a sort of a risk benefit understanding, that why you want to investigate and what is the benefit that the subjects or the investigation will get you to. Next slide. I'm not going to go into all of the regulatory and administrative components. These are all in the FDA website. You can obviously know how to, and we help sponsors put together. The important part is the middle part, the high-level clinical development plan. The agency often wants to know that you want to do the first in human study. Now, it's just not the study, but how does that study fit into a clinical strategy? You know, how does it fit into? What kind of information you are getting from that particular study? That is going to be helpful within the context of the broader clinical plan. I think that part the agency always tries to understand, you know, because that's very important. I mean, so that, you know, they understand exactly what you're trying to do. And again, I mean, you may be successful and you may not be successful, and they understand that. But it has to fit into a story, for example. Next slide. And this is my last slide. The review phase. The review phase is a 30-day clock, as you know, and this is this is applicable to the initial IND. After 30 days, FDA will give their action. Either they will say you may proceed or will be placed on hold, and the hold can be partial hold or it can be a full hold. Partial hold, sometimes we see that FDA, you may propose that you're going to try 4 doses. They might say based on your tox data or based on other information, they might say you can only do the, you know, the lowest 2 dose, and then you cannot go to the highest dose because you do not have enough safety margin, for example. You have to do other animal studies to be able to proceed to the higher dose. So sometimes we call that as a partial clinical hold. Within that 30 days, there are opportunities for discussion. I know, and oftentimes, you know, the time is short. As a reviewer, a lot of times, and now probably in the last 8.5 years, while I was outside the agency, the reviewers probably get a little bit more time, but we used to get actually only 2 weeks before it comes to, you know, the package is available. It's really rushing to get all of this information and try to review, particularly from a safety perspective. So there are some opportunities for back and forth. Agency might ask for information request or make some additional, you know, you might have to do some additional submissions. But after 30 days, they will take you know, either action, which is that you are, you know, you are safe to proceed. Oftentimes you will also get a lot of different comments on your program, which are not hold comments, but those are kind of a recommended things that you should be doing. So that's, I mean, they try to be collaborative and help you as you move through your clinical development. So I will stop there and hand it over to Mo. Thank you.
Good. Thank you so much, Partha. Today I'm just going to talk a little bit at the end about cell and gene therapy, which is likely to be considered the fastest growing part of the biotech industry in the last few years. I worked in this space maybe 20 years ago. In fact, I was working in Research Triangle Park in the Becton Dickinson technology at that time, a long time ago. The industry has evolved and has matured and has produced some success in recent years, and it's expanding quite rapidly. And I know there is a ample amount of activities in Research Triangle Park currently in cell and gene therapy space. So I'm very pleased to be here. This slide basically just highlights what everybody knows, which is the expansion of clinical trial opportunities in this space, which are focusing in, you know, different buckets: gene therapy, gene modified cells, cell therapy, and tissue engineering, which encompass combination product. And the primary indication in this area that is focused on, obviously, as you know, is oncology, but there's an ample evidence and expansion of those activities in neurodegenerative conditions, metabolic disorders, musculoskeletal, and ophthalmology. So we can go to the next slide. The next slide sort of summarizes a general trend in the space. We know there is a huge amount of growth in oncology and inherited genetic disorders. Monogenic diseases are being addressed by gene therapy products quite successfully. Most of the product in this space qualify for orphan drug designation, which brings a huge amount of incentives for the developers in US. There's obviously regulatory framework is very complex, but US FDA has been very flexible and collaborative in working with the industry to accommodate some of the major challenges that are seen in this space. And as you know, there's a shift from a small mom and pop show to larger pharma, and the larger pharmas have really adopted this space of cell and gene therapy. There has been a lot of big acquisitions in terms of tens of billions dollars based on one molecule, based on one therapy alone. So there is a significant amount of interest in the industry from pharma and biotech. Again, there is flexible interpretation of existing regulation. Existing regulation has not been changed, but there is a flexible interpretation of those existing regulations, which are articulated in form of a guidance document, etc. And the way FDA reviewers interface with the community through outreach activities, presentation, etc., and so forth. There are preceptiveness in innovative trial design, single-arm studies sometimes is acceptable in oncology space. A small patient population can be treated, etc. And of course, there is a high risk, but there's also high benefit, and those high risks have to be mitigated. Additionally, in this space, cost is a factor I wanted to highlight, and manufacturing remains to be the major hurdle going forward. In fact, most of the clinical hold comments from FDA, whether it is at the IND phase or post-approval or at the level of approval or acceptance of the BLA, are based on CMC consideration and manufacturing issues today. So I want to highlight the importance of that. We can go to the next slide. Before going there, sorry, I wanted also to stress the expansion in the number of INDs being submitted to FDA. The chart to the right highlights this expansion in gene therapy, which is exponential, versus as a cell therapy, which has been predominantly seen within the agency. So there is a shift from the purely cell therapy products to gene therapy space as we go forward. And the size of Office of Tissue Advanced Therapy, and as I will mention later, the review branch has doubled in size in the last couple of years. There is a significant number of new reviewers that have joined the agencies. And of course, with any sort of a new newness in this space, there's obviously some challenges in training and getting the FDA reviewers up to speed in a very sort of limited time that is available. So I just wanted to stress the importance of how dynamic this space is, how some of the activities have accelerated in the recent years, and also emphasize that there are challenges that really centered around not only regulatory hurdles, manufacturing, but also from FDA perspective to be able to review all of these submissions in a timely fashion with the highest quality available, using the best of the knowledge available to the field. And those challenges are remaining and are being addressed by FDA as well as the industry going forward. So the next slide, we can deal with the different products that are offered in this space. This is an important aspect of cell and gene therapy. The products are hugely diverse. We can deal with a number of different products which are outlined here. Traditionally, there has been a lot of success in the approval of a cell therapy product initially at FDA. products such as Cardicels, Provenge, Laviv. There have been like up to 8 or 9 cord blood banks have been licensed. Then recently there has been a shift to gene-modified cells, the CAR-T product, Kymriah, Yescarta, and others have been approved in this space, which are gene-modified lentiviral vector or retroviral vector transduced cells. There was one oncolytic virus product that was approved by FDA as well a couple of years ago. And then recently we are seeing the gene therapy products, which are based on AAV vectors like Cellular and Zolgensma, that are approved by FDA as well. This slide, I just want to go through some of the component of this slide to highlight the diversity of the product and the complexity that really makes the review process very difficult and challenging, as well as submission of these documentation to FDA. Of course, FDA is really an Office of Tissue Advanced Therapy. I will talk about how it is organized in a little bit, but the amount of knowledge that is needed to cover all of these diverse products is quite challenging, and that's something to be kept in mind. There are some products that are purely cell therapy products. You may be familiar with MSC products in this space. You have products that are gene-modified cells, such as CAR-T cells and some other products in this space. The third category includes gene therapy products. These are products that are essentially fundamentally driven by gene replacement strategy, and the common vehicle for transfer of the missing gene is AAV vectors in this space. And so there's a number of products are being developed using adenovirus associated viruses as a vehicle for delivery of the missing genes for monogenic diseases. Then we have another category of products which are called gene-edited cellular products, as you heard about. These are the products where specific genes are edited, the point mutations introduced. There are a number of editing is introduced to the cell and ex vivo, and the cells are actually are the products that are given to patients. Of course, this is commonly referred to as ex vivo gene editing. There are up to 10 to 15 INDs in ex vivo gene edited products at FDA. But recently there has been also activity in area of in vivo gene editing. And in vivo gene editing, which is not described here, is essentially the concept that gene editing agent is delivered to the patient directly through a AAV vector or other means, and the gene editing occurs in vivo. And of course, you can realize that in vivo gene editing would be highly complex and much more complicated as ex vivo gene editing. And there are a number of things that has to be addressed. And then we have combination products where biologics and device are combined typically, and this area is growing as well. These are considered tissue engineering products essentially. And of course, we have secreted products such as exosomes that are being handled and regulated by Office of Tissue Advanced Therapy as well. Therapeutic vaccines, such as therapeutic peptide vaccines, are regulated by Office of Tissue Advanced Therapy, where I work, and xenotransplant products are also regulated as well. As you know, there are a number of activities in this space in gene editing of animal and pigs to allow xenotransplantation to occur without immune rejection. And also gene editing has been incorporated in a number of these platforms going forward. And there are a number of other products that constitute advanced therapies. I wanted to highlight the differences of these products and to give you that perspective that within office of tissue advanced therapy, all of these products are being regulated, and the know-how exists in that office. If we can go to the next slide. This kind of highlights essentially why I think cell and gene therapy is much more complex than any other product from my perspective. This gives you a highlight of the differences between cell and gene therapy and biotech product. Biotech products are highlighted in light blue. The major issue is the manufacturing of the product using master cell bank, producing monoclonal in a consistent fashion. But for cell and gene therapy, obviously, there are other considerations to keep in mind. Collection of material becomes a huge important aspect. The donor eligibility with respect to allogeneic product is very fundamental because disease transmission is a major source of the safety concern in this field. Of course, the transportation of the collected material to the manufacturing facility is another consideration. And then, of course, the overlap would be that there is a overlap with biotech product in regard to challenges in manufacturing. And the final drug product obviously has to be formulated sometime, further manipulated at the clinical site before administration. And there are novel devices that are being used for delivery of some of these therapeutic. So we have some major challenges in dealing with the different aspects of the cell and gene therapy, looking at from the perspective of collection that occurs in a non-GMP environment, transport of the collected material, further processing of the incoming material, and then manufacturing of the product and the delivery to the patient. Next slide. This sort of gives you a perspective of Office of Tissue Advanced Therapy in the Office of Director. There are three divisions. One division deals with the product. This is a division of cellular and gene therapy. The other division deals with the clinical and non-clinical consideration, DCEPT. And then the third one is human tissue. I want to emphasize the importance of this division. This division solely looks at the compliance of the allogeneic product and human tissue in regard to disease transmission. And they have a number of reviewers that look at donor screening and testing and look at the compliance and look at whether the INDs are in compliance with donor eligibility and screening if applicable. I also wanted to stress the type of products which are regulated. The 351 products are well known. These are drug products. Again, the regulation that are applicable for these products encompasses some of the regulations that are applicable to all drugs. I wanted to highlight, if you can see, the importance of being aware of the applicable regulation. For example, you can pick up one of these booklets that deals with, for example, regulation 210 and 211, and I don't know if you can see it properly. I encourage you to pick up one of those booklets. The other applicable regulations are 600, 21 CFR 600. These are the regulations that are applicable to biological products. So all biological products are drugs, but not all drugs are biological products. And the last one, which I will highlight, is 21 CFR 1271. So please go ahead and buy these three booklets and read at night all the regulations that are applicable to biological products. It's very important. It's an important read for you, and you become familiar with all aspects and the regulation. All of the regulations are driven by science and the technical knowledge that you have to have before administering the product to the patient. So for drug product, you have those three applicable regulations, right? And then there is another category, which is called 361 product. These products, they do not need to go through pre-market approval. You don't have to submit an IND, but you have to register the facility, and if applicable, you can market those products if you comply with certain regulation as outlined in 21 CFR 1271, subpart C, and good tissue practices, which is subpart D. Obviously, these facilities have to be registered. A large number of stem cell clinic today, or as well as other tissue processing center, want to say that there are 361, there are marketing 361 product, but sometimes FDA disagrees. If you are manipulating the cells in a way that is not permitted, if you're doing more than minimum manipulation, if the use is not homologous, then FDA essentially says that you are marketing a drug product, and because you have not submitted IND, you have to cease marketing of those stem cell products. So some of those things have been publicized recently in this field. So a lot of these stem clinics are being inspected by FDA, and they've shown not to be able to essentially release those products without approval because they really are categorized as drug products, not as 361 products. So we have 9 minutes to go, couple of more slides. The next slide essentially talks about what Partha alluded to, which is how the INDs are reviewed. In Office of Tissue Advanced Therapy, obviously the same regulation applies. You have 30 days to review the IND material. Most reviewers within a couple of weeks can provide you with a list of their questions, which is outlined in information request, and I encourage you to rapidly respond to those information requests as much as rapidly as possible to allow them to give them enough time to assess that information. If you provide the information late, most likely you will be placed on hold if the information request really outlines something critical to the manufacturing. But if you don't hear by 30 days, you're allowed to proceed. And if you are notified by a project manager, essentially you're notified that you're on clinical hold. That is not a good conversation to have. Again, as shown in this slide, the way INDs are reviewed is essentially assessment of the benefit to risk ratio. And the consideration for that is outlined in this slide as well. One consideration is the type of product that I didn't mention here. Obviously, the type of product defines benefit and risk ratio as well in view of the indication of use. The second consideration is the safety of the product, and how comprehensive you have been in establishing the safety of the product in vitro and in animal model study that allows you to proceed with human studies. And the third principle that is used is a risk and science-based approach. Within OTAT, the science plays an important role, and in your discussion with the agency, I encourage you to use a science-based strategy as much as possible, mix with the risk-based approach to have a very fruitful conversation with the scientists in the team. Institutional knowledge is also very important, and in some cases, there is a huge amount of institutional knowledge. And so the reviewers are aware of things that you may not be aware of as you submit your information. That institutional knowledge helps you really assess and be able to deal with some of the issues that you may consider. The whole process is a team approach. The preclinical, non-clinical players and clinical reach out to the CMC folks quite extensively and have a discussion about the product as it relates to preclinical study and the safety profile and how it relates to clinical study. And everything is case by case, obviously, as you know, because every product is very different from another product. And so case by case prevails all the time. Next slide.
I'm a little conscious just of the time. Some people might have to log off. So before we go through that final slide, I would like to invite the participants who have any questions now, maybe to ask them. If anybody needs to log off right away, you know, those who can stay a bit longer, we can proceed as well. Judy, you've been collecting some of the questions. Please go ahead also and maybe share some of the questions the audience has already. Yeah? If not, we can also have some of the questions we also have.
I have a question from Andrew Prince, and it says, beyond drugs, will you be able to discuss devices more specifically when folks from Parexel reach out for the office hours?
I can answer that one, Partha, if you'd like. The answer is yes. We do have several SMEs that are dedicated and are ex-FDA reviewers from the device divisions. So yeah, the answer is yes.
Great. I think that there probably are other questions coming in, but perhaps we can ask some that we've sourced from the companies that have reached out to us, Miriam.
Yeah, so and I have a question from one of the gene therapy companies, you know, which is what causes something to be classified as a combination product, you know, for example, a viral bulk vector plus a protein used together. Is it a combination product? Is it formulated together? What if they're delivered separately in a short interval? Huh?
Yeah, so I think Partha can also answer that question, but combination products is really looks at if the combination is between a device, biologic, or a drug. So if you have two biologics, it's not a combination product, right? And if you have a device and a biologics, you have a combination product. So often the discussion is around if you, during the manufacturing of this gene therapy product, you use a biologic, that is not a combination product per se, because during the manufacturing, you're using biologics to make a product. The final product would be a biologics, but you may use a small molecule during the manufacturing. That doesn't constitute the terminology of the combination. So the example of the combination products that are well known is if you use MSCs in conjunction with synthetic scaffolding material, that would be a device biologic combination. If you're using a AAV vector in combination with a device for delivery, that would be a combination product.
And another question is, will the FDA audit preclinical facilities before approval of the IND? Is it mandatory?
No, I think Partha, you can.
No, yeah, no, and the answer is yeah. I mean, no, usually, I mean, the FDA doesn't have that many resources to do that. Oftentimes, you know, they will do some inspections if that facility has been used multiple times for other drugs and things like that. So there is a routine inspection for some of those things, but really specifically, again, that's a risk-based inspection, and it usually will not do that. Yeah.
I want to just say, I know you had some question around this of how CGMP is applicable in cell and gene therapy or the space. Obviously, FDA has a phase-based approach. For initial phase, safety is very important. Compliance with statutory CGMP is more important. Full CGMP is not applicable during the initial phases. As you go forward during the development, the CGMP becomes more applicable, and by time of licensure, you have to be in compliance with full CGMP. So we have a phase-based approach. I want to stress that importance because in cell and gene therapy, quality of material initially are not clinical grade, say, in some situation. So it is possible to have research grade, but you have to determine the quality, you have to verify quality, and because that is a safety issue. And the FDA reviewers look at that quality of the material very carefully during the initial phase because clinical grade or CGMP grades are not available. So that is the flexibility that exists.
And is that not the case for other non-gene therapies? So if you have other biologics or just other drugs in general, would they have to adhere to GLP and GMP during the IND application?
Yeah, I mean, so if I can take from a drug perspective, oftentimes, tox studies, for example, or a safety pharmacology studies, those are usually the expectation is GLP, because that ultimately defines the safety of the product when it is first used in the clinical trial. A lot of the other pharmacology type of studies and all that, the strict GLP is not needed. But again, it all, as Mo pointed out, agency will look into that very carefully in terms of the quality, the consistency of the product, and manufacturability of the product at that time. Again, depending upon how much you are manufacturing to support what kind of trial, that becomes also very important. How many subjects you are exposing that to those lot, for example. So yeah, so all of those things come into play. But yes, the thing to remember is that any types of non-clinical studies, animal studies that you're doing, it has to be GLP if it is something to do with the tox, the safety.
Great.
And I just quickly, for cell and gene therapy, that is also applicable, but just keep in mind some of these models don't exist for some of these, indication or intended treatment or mechanism of action. So agency has exercised flexibility. The official wording is that, yes, you should perform GLP studies, but if there is justification, if those models are not available, if they are going to obstruct development, they actually listen to you and look at it from perspective of risk and science. That's why science is very important.
And then kind of like a related question from the audience too, can we use a different batch or lot of test materials in the GLP toxicity studies for the IND submission studies?
Yeah, I mean, I can say that the agency's advice is to use as similar as you can get the product for GLP studies or tox study as you would use in clinical setting. So the product has to be as similar as possible, although sometimes it may not be identical, right? So there is some flexibility from that perspective in the cell and gene therapy space. I think partly you want to comment on the small molecule part as well, if you like.
Yeah, it's the same philosophy. I mean, it's essentially the same philosophy, right? So yeah.
Yeah, they'd like you to treat the patient with the same product you use in animal studies. That's sort of a common sense, right?
Right.
And for those who are looking to, I mean, as they're, kind of thinking about scaling up, their manufacturing, obviously many have this small facilities, even if they try to adhere to GLP or GMP, if it's a small batch, and as they scale up, what are the recommendations on approaches they should take? Yeah.
Yes, so in gene therapy or cell therapy, that's a common occurrence, right? Most of these are developed in academic centers, small scale. They show a lot of promises. All of a sudden, you have to do a pivotal study. But you think about commercialization and do a scale up or scale out, whatever the case may be, which involves manufacturing changes, demonstrating comparability, demonstrating that essentially that the new manufacturing facility can make the product that is comparable to product that was made previously. So the couple of aspects of it is, in most cases, there's a shift from the small academic manufacturing facility to well-known commercial manufacturing facility in this field. Most of the products are being made by large CMOs. And in some cases, if there is sufficient funding, the company can choose to establish their own facility. But that is not typically how it is done, unless it is supported by a large pharma, etc. So being able to make the product reproducibly, consistently with high quality that is similar to what you manufacture during academic stage and in your facility is critical, and that has to be established in the process. So Part of it is comparability, tech transfer, identifying the right CMO, being able to verify they can produce high quality, having conforming batches made by them, etc. So you have to think about that and plan for it.
And as our companies think about this and try to engage with CROs, CMOs, what are the things that they should be looking for, in hiring a service provider, anything from in terms of communicating with them, is it better to use a smaller niche company, a big established one? How do they know they're the right?
Just hire Parexel.
Yeah, exactly what Parta said. But Parta, it's very complex. Joking aside, is like part of it is vendor qualification. You have to do. You have to perform audits. You have to look at the compliance history of the CMO. The more compliance history they have, if they have been already inspected, of course, it's going to be advantageous, correct? And so the compliance history, whether they comply with CGMPs, just because somebody says they are CGMP compliant, that is not so unless that compliance has been verified by FDA or verified by Parexel. That one can do an audit of the manufacturing and say they likely to be complying with CGMP. So those are the aspects. And their manufacturing history, what they are good at. There are gene therapy CMOs that have only done gene therapy products. There are some that are cell therapy that have done, and there are others that have done gene modified cells, such as CAR T. Each one of them has a skill set and a niche area that they excel in. It's hard to be good at everything.
And then one question, I'm just conscious of the time and the audience. I know a lot of have stayed, but and I want to just say final comments after this last question. You guys mentioned, Partha, about engaging with the FDA initial stages, and you recommended that. But when should they really begin? Like, what is that? Are there any drawbacks of engaging them too early in this product development process?
Yeah, so obviously, let me address from a drug side. From the CBER side, you have that interaction opportunity as an interact meeting, which is informal and very early in the game. For the drug side, you have to do the pre-IND meeting. Now, the question is, if you do too early, think about, put yourself in FDA, and say, well, do I understand everything that you're trying to do? Yes, broadly, I understand what you're trying to do, but I don't know a lot of the information. So going it's too early, sometimes you are like, because you're not going to go there and tell FDA that, can you, help me develop the clinical development plan or product development plan? You have to develop that. And then, show it to the agency, and then you have to tell them this is what you're planning to do. It's not that you have to have all the information, but at least you have to have the product and some level of characterization, or maybe some mechanistic information that yes, the product has an opportunity to work. And then you basically say, hey, you have to kind of show them the roadmap. This is what you're thinking, and that thinking may not be completely fully vetted out, and that's okay. But you have to, show them that this is what you're thinking, and then they can react to that, respond to that particular plan. But you're not going to go there and basically, show them all of the information and basically say, help me out here. That's usually will not work. If you go too early, the agency might come back and basically put a very nice introductory comments, and they're probably not going to answer some of the question, and basically will say, see the introductory comments. Maybe there is a big flaw, or they will say you do not have enough information to be able to comment on that right now. So those kind of interactions become less meaningful, but hey, there are things which you saying, what, I do not have to ask 10 questions. I have only 2 questions, which are extremely important, because if I don't get an agreement or some kind of assessment of what they're thinking, I cannot move forward. And if that becomes an important, then it's obviously you can go and have the discussion.
And so I just wanted to emphasize that CBER has 2 informal programs, as Partha alluded to, Interact and the CAT, which is another one that looks at platform technology. To keep that in mind. Those are informal. So you can be often denied if you don't have appropriate information. Pre-IND is a formal meeting, and often you can get pre-IND meeting with written response. So that I just want to keep that to highlight that as a difference.
Thank you. Just to close those remarks, I would just kind of want to ask both of you, if you have, what is the most critical piece of advice you would give one of our early stage founders, you know, who's transitioning from this very early stage proof of concept studies to IND enabling studies? You know, what would you say to them? Yeah.
You have to, I mean, I can step in and say, you need to know the drug target profile that you mentioned. That was one of your questions. Mm-hmm. You need to define it early on, and you need to have a science-based approach. Identify all risk associated with the product and try to mitigate it in vitro and in animal model. And if you do that, you're successful in your IND submission.
Yeah, and just another comment on to that, and I agree with Mo on that. Just on an additional from a business perspective, you probably want to find out, you know, try to see which are the patient population you're targeting, and probably you'll have some assessment of the market, you know, where the market is moving, where you know, what are the other competitors in the space. So you need to figure out, you know, 5 years down the road, or 7 years down the road, 10 years down the road, how does the market look like, and which are the patients you are bringing the product to. So you have, and that is kind of related to the target product profile. It's a both of a regulatory risk, but also a business risk, and also the access. So all of that combined together, I think is going to be important. I mean, again, you don't have to know everything, but you have to have some kind of an idea why you are developing this.
Thank you so much. I mean, this has been very, very useful, and I think a lot of our audience is going to be left wanting more. So I think which is a great place to be in terms of this initial contact with you. We're very excited to keep working with Parexel and offer this office hours to the startups and faculty from UNC. I'm sure we will get a lot of interest from this. So I just wanted to thank you again for a wonderful talk. And if it's okay with you, we can share the slides with the participants.
Sure, yeah.
Sure. All right, thank you so much.
Thank you so much.
Thank you.
Cheers, bye-bye.
Take care,
Bye-bye.
bye-bye.